Okay. Thank you, everyone, for joining us. My name is John Sosa. I am the CEO and co-founder of MIPAR Image Analysis. Joining me up here at the podium later will be Paul Sewell. He is our senior application specialist and has a very strong biomedical background. So about halfway up through the presentation, I will invite him to go through even more case studies of challenging bioimage analysis problems that MIPAR has been able to solve. Thank you very much to Rushing for organizing this great workshop and inviting us to attend. We've seen a number of great talks so far, and I really appreciate the opportunity to introduce you to MIPAR, which has been very exciting for us. So, to introduce you to MIPAR, it started at Ohio State. So I was the primary developer on it at Ohio State University, where I got my degrees, undergraduate and PhD in material science. It's been under development since 2007. It started in material science, as I mentioned, and it's important to note that it has been and continues to be developed by actual users. I'm not a software developer by trade, nor are any of our developers on our team. We are all end-user scientists that built this product for us to use for our own research and continue to use to help with other people's research. Even though it's been in development since 2007, iteratively, of course, with using it, we spun out as a private company from Ohio State just last July. So I think about 8 months in now to our first year. In that short time, we are very proud to be able to list major companies and universities as just a few of our key customers. In total, in this time, we've amassed about 2,500 users in over 40 countries around the world. A number of these users had never had any experience with image analysis prior to MIPAR. Many of these users have, and they've told us, happily switched from ImageJ, from Python, from other open source tools. And many of these users have switched, as they've told us, from other commercial solutions that are out there. So, where did it all begin? I did my research in titanium alloys at Ohio State. And titanium alloys are an interesting animal. They have very complex features under an SEM, and they can look all sorts of different ways depending on how you heat treat them and alloy them. So we were working on predicting how alloys would behave mechanically based on what their microstructure looked like. It was the beginnings of machine learning predictive models before that term was hot like it is today. So we needed to quantify objectively and automatically various traits of this complex system. So we built a tool that enabled us to do that. It would do things like this: complex segmentation of various features in one algorithm. These algorithms we call recipes. We've been calling them recipes for some time. So it's actually quite nice to see that term, as we've heard it a number of times this workshop, catching on and being used more and more. But these recipes are algorithms that are visually built with no coding in the software, and they allow you to do some very complex segmentations very efficiently. Why? Because, as we all know and we've heard, your measurements, which is the whole reason you're doing any of this, are only as good as your segmentations. So if you want to measure the size and shape of these purple bits, you better be selecting them and separating them properly. The types of measurements that we can get out of these are all over the place. Global measurements like the amount of the red phase, the amount of the green phase as a percentage, the volume fraction of the purple, the particles and the plates, the diameter of the particles, the thickness of the plates, Once you have your segmentation, you can pull out all sorts of measurements. So this is where it started, and then we moved on in materials. And we've handled challenging applications like fiber detection and thickness. This one I love because behind the fibers is just about the same gray value as the fibers. So this is not trivial for most image processing to go in and find what are the fibers versus what are the background. We do that with a recipe. Once you have the fibers, you can then do all sorts of measurements again. Here's an example of local thickness visualization. So the fibers colored according to how thick they are, with a histogram that's color-coded the same way. Yellow is thick, blue is thin. We've got things like grain size, patches of material that have different crystal orientations, as it's known. You need to detect the boundaries between those patches. Those boundaries can be identified with a thin black line, or in the same image with no thin black line. You've got to have an algorithm, if you want to automate this, that can work with all of that complexity. You've got the grains found. You can measure their size, their shape, color code the features by their size, by their shape. You even have a nice interactive table over here with all of the sizes and shapes listed, and when you click on a cell, that same feature is highlighted. Or when you click on a feature, that cell is highlighted, and you get a line in the histogram where it shows you. So there's an interactivity to exploring your measurements in MIPAR that we really like and our users really like. And then most lately, we've been working with NASA, and they are exploring a new standard to quantify particles in a high-temperature alloy that's used in aerospace called nickel-based superalloys. They are trying to promote this technique using their imaging method and our software as the new standard for how to measure these critical alloys in jet engine components. So we're very pleased with this partnership. Here you can see we've sorted the particles by size. Green is small. We call them tertiaries, secondaries, or the red, the bigger ones. So these applications, the need to solve these is what led to MIPAR being MIPAR. So I took a stab at a slide to efficiently describe what MIPAR is. If we had to boil it down into 3 clauses, it would be a visual algorithm builder, absolutely no coding required, that enables rapid creation of custom automated solutions that work, which can be supported easily by experts, either us or experts in your group, in your lab, especially if they've had time with ImageJ. That's a boot camp that's going to be great when they get started with MIPAR. If I had to break down now this into a Venn diagram, I would say that the visual nature of MIPAR, the intuitive design that we really put a lot of time into getting right, gives everyone access to the following: the flexibility of coding without coding, the efficiency of drag and drop, both in building the algorithms and running those algorithms on your images. That again, because of these 2 things, because you can rapidly build flexible solutions, we have a business model that lets us build these for you quickly. And your experts can build them quickly for the rest of your team, or teach them to, because learning curve is doable. So that is how I would summarize MIPAR: a unique balance of flexibility, efficiency, and support. And it is just by accident that our white logo makes the same shape as the intersection of, I couldn't help myself. So I know that this seems maybe pie in the sky. Who doesn't want a software that's these 3 things? But if you let me show you, and Paul show you, what we've been able to do with this, I think you'll see this is for real. So we're going to take a journey through various case studies of MIPAR being used to solve real-world, challenging bioimage analysis problems. We're going to start with a problem from our valued partners at the University of Louisville. They had a wound healing assay application with a lot of images. And mostly they had the same imaging conditions, but not always. And they needed a solution that let them, instead of manually tracing, which is what they had to resort to, find the wound areas and measure them. So this is the interface of MIPAR's image processor. This is where recipes are made. This is where they're executed on single images. And it's where measurements on single images are made. Users have told us that it is the cleanest, the simplest, and the most intuitive image analysis interface they've ever worked with. You have your reference image on the left, which is your original, and then you have the current, before and after, the current state of the processing. You can update this with a new image in your processing sequence, but by default it's the original and the current state of the processing. To run a recipe that, again, is built automatically in MIPAR, you drag it in. So here's our wound healing recipe. Finds the wound in this case. This recipe is completely interactive and non-destructive. So you double click on a step, it shows you what that step did. Here's where we've set a threshold. Here's where we have kept the largest feature in the threshold. Here's where we've removed features under a certain limit just to set a minimum wound size. That didn't do anything in this case. And then we filled in holes. A pretty simple recipe. What do we think the chances are it's going to work on the next image? I'm going to say it's doubtful. But we can try it easily. You drag in the next image, the recipe automatically runs on it. Didn't work so well. What we can do is fix this quickly, make the recipe more robust, learning from, now we see a different condition that has variation. First thing we're going to do is we're going to add a pre-processing step at the very top. We didn't have to duplicate our image at each step. We don't have to undo the last 4 steps that we did. We go to the top, we can be looking at the final step, we click once on the top, and we go up to our pre-processing menu and we open a contrast adjustment tool. We're going to set it to auto mode low, which is a black balance basically. It sets the lowest value in the image to be pure black. You've got preview on and off down here so you can toggle it, see what before and after looks like. Every module has a help button that takes you right to that page in the user manual. You say accept. That step now becomes the new step 1. It gets inserted at the top, and without you doing anything, the recipe automatically rebuilds. Now you see the updated final result. It didn't change in this case, but that step makes this recipe more robust because overall it sets the intensity range of the image more consistently for images going forward. Now we're going to add another pre-processing step. So the reason it didn't do much is because while it overall adjusted the intensity of the image to be similar to the previous, there was a relative difference going on between the brightness of these cells and the brightness of this wound. Moreover, or in other words, intensity of the original grayscale intensity of the image is not enough to reliably separate wound from cells. There's texture information that's going to come into play. So you go to the top. Now we're going to insert, you just select the top step, go back to pre-processing. We're now going to pick our bright texture filter. This is going to light up areas that have bright texture. That's the before, so that's the adjusted contrast result. Bright texture does that. It keeps or even brightens the cells and keeps the texture-free patches dark. Again, that step gets inserted and the recipe updates. If you're like me and you've struggled with duplicating images just so that you don't lose all of your work when you have to make a setting, you'll understand how critical that aspect of MIPAR's design is. There's no writing down steps so that you can make a macro later. Experimenting and building your algorithm is building the macro. When you're done, it's done, and you click save. We've got people building macros without knowing it. People have told us they've hated building macros, and by the end of our presentation, they're asking us, is there a limit to the number of recipes that we can make? We've actually inspired them to want to make these now. So, the last thing we're going to do is we're going to modify the threshold step. This allows the user to set what they call the wound. It's a subjective decision. You could draw this boundary wherever you want, but the value of automated image processing is you set the rule once, and it's always going to run the same way, whether you've had your coffee that day or not, or whether you give it to the next person to run. So you click on basic threshold, you edit down here, you edit that step, the preview pops up. What we're going to do is we're going to keep the auto threshold, but we're going to offset it. So it's going to say auto threshold level minus some offset value. We're going to reduce it by 40. And that is what this user wanted to define the wound boundary. So we click accept, that step is updated, and automatically the recipe rebuilds, and that's now our final wound detection. We do have ways to let MIPAR objectively choose critical settings like that, and I'll show that in the next example. Call it optimization. You gotta go back, it's actually this button up here. If you're on a step and optimize lights up, that means you can have MIPAR tell you what its settings should be. Most of the critical steps are optimizable. So, we save the recipe as a file. It's probably 7 kilobytes. You can email it to whoever you want. And now we're going to go back to the first image. We drag it in. So it works well on this image. Recipe loads. Works well in the first image. Real test. What does it do when you've got no wound? Drag in an image with no wound, and it works well on that too. It comes up with nothing. So, what have we seen? Honestly, from our experience, 6 steps is a pretty simple recipe. These are not black magic tools going on here. What we've seen is that with the right interface, very simple tools can give you pretty robust solutions. And they can do that if you design the product efficiently and allow it to efficiently make these robust solutions from these simple tools. You're able to get places more quickly than you can with some open source things that we've seen. And you're able to get places that even with all the time in the world, you may not have gotten to because we're humans and we have patience limitations. And the ability to visualize what you're doing as you're doing it opens up some creative insights that are intangible. So in the biospace, all of this gives us a tool that has flexibility in the presence of real-world variability. And I'm new to bioimage analysis applications, but they are some of the most varied, the most complex that I've encountered. And you've got to have a tool that matches that. So once you have your recipe, we've got a batch processor. You can apply it to thousands of images at once. How does it work? You drag in your recipe, you drag in your images from wherever they are, and you click go. Once it's done, you can review how the batch actually worked in our post-processor without blindly making measurements from your batch without looking at it first. We call it quality assurance. So, this is our post-processor. Segmented images up here, originals down here. You can overlay as an outline, as an overlay, as the binary, as the individual features colored differently just by changing this drop-down. That is something we now as developers often overlook. Not sure how many steps it takes you to make an outline in image J, but if it's more than one, it's too many, in my opinion. So, post-processor, again, I don't highlight it, but that's where the segmentations live and the originals live. Something we just added in our latest release is the ability to save and resume sessions. So if you've loaded in 1,000 images and you've got multiple layers, layers are what we call different features being segmented. So you might have one layer that would be the wound, another layer could be the cells, Another layer could be some other feature in here. You can have as many layers as you want. You have multiple images with multiple layers that you've already set up and started reviewing. It's very nice to be able to save this as a file and resume it at any time. That's something new that we added. We call them sessions. You can output all your segmentations in whatever viewing mode you like with a click up here. You can manually clean up very efficiently image after image with our image manipulation tools, common outlining, ellipse, box, freehand, paint bucket, all the things they're used to. You click through the images, you make a change, you click save, you go to the next one, you make a change, you click save. It's facilitated manual cleanup that is a great time saver. So we can click through. Here are the first 3 wound examples we saw. We have more, and the recipe performed nicely. At least that's what our user told us on all of them. Now we want to make measurements. We'll make global measurements in this case. We'll measure the fraction of the wound that it occupies in the image. So you go down here to measurements, we check area fraction, and we check and we click generate. And we get a table, image number, area fraction of the wound. This agreed with what they thought it should be. You can copy and paste this into Excel. You can save it as a file. That's batch global measurements in 2 clicks. So that is a common workflow that we and our users use for 2D analysis in MIPAR. You've got the image processor where you build and optimize the recipe, the batch where you run it on many images, and the post processor for quality assurance and batch measurement generation. So the next study I'm going to show you is more challenging. It involves more classes. This was a collaboration we did with a user in the microbiology department at Ohio State University. It's been a fantastic project. Here's what he had. He had a fungal assay. This is not initially what it looks like it is. This is not bright objects that you just need to select and count. I wish it was. These are fungal colonies, as he's taught me, that have different degrees of filamentation. Probably can't see it with the projector in the audience, but around these bright centers are these fuzzy rings. And the extent of those fuzzy rings tells them something about how much the fungus has filamented. And that tells them something about something that's important to them. That's as much as I understand it. There are some colonies that have not filamented. I think there's one down there and there's one down there. It was critical that they catch those and count them and express the number of unfilamented ones as a percentage of all of the colonies. So this guy is a pro. He knows Python in and out. He knows ImageJ in and out. He was in the audience when we gave an introductory talk to the microbiology department, and afterwards he came up to us and told us his story. He said for months he's been, and he had clever ways of writing scripts in Python and running them in ImageJ and vice versa. I didn't know you could do those things. He said for months he's been struggling with this and he was not able to find an automated solution. So their study was bottlenecked by the analysis. They were manually scoring these things, and they could do, I'm not sure what the number he said was, a handful a day. So the bottleneck was the scoring. He told me then something I've never heard during a live talk, which is that during the talk, I guess maybe he wasn't paying attention to all of it, because he had supposedly remoted into his workstation back in his office. He had downloaded and installed MIPAR, and he had built a recipe to automate this solution while I was giving the talk. I couldn't believe it. So this is what he put together. It's a longer recipe than the first one, and you'll see some new features, but you drag it in, and it detects the colony centers and the fuzzy rings around them, and the colonies in blue that are free of filamentation. You notice, looks a little different over here. We've got what we call chapter headings. which are infinitely customizable. They basically break the recipe down into plain English sections. This is the section of steps that does pre-processing. This is the section of steps that roughly finds the colonies. This is the section that cleans them up. You can name these whatever you want. You can even, as Trevor mentioned, name recipe steps whatever you want and give them custom notes that other users that you share the recipe with can see. You can call threshold the, super proprietary object detector if you want. You can name them whatever you want. So chapters break up the structure and make it easy to read. The colored steps are layers, as we call them. So at the point in the recipe where the centers are detected, you set that as a layer, you give it a color and a name. You do the same thing for the filamentation and the colony free of filamentation, and now you have a new viewing mode up here called layers that when you select it just shows you the steps that you've set as layers, color-coded the same way as they are in the recipe. You can set as many of these as you like. There's benefits to measurements. When you run measurements in layers view, it runs on all of the layers at once, unless you want to limit it to just a few. Just a few of them. So those are layers. With a switch of a drop-down down here, you can switch this to outline mode, and this shows you some pretty impressive things, quite frankly. Colonies like this, watershed alone didn't do that. There were some other steps that had to be done to separate that colony with that thin line, but all of these subtle colonies are properly separated with this recipe. The separations pass through both the center and the filamented area. So I'd like to run through now some of the critical tools that were used to build a recipe of this nature. I'm going to start with spot removal. It's like spot healing in Photoshop, but automated and able to be applied across an entire image. It's a lot of how we get rid of artifacts before they have a chance to ruin our lives. So, as an example, we had these bright spots in the center of the colonies. This threw off a lot of the segmentation, a lot of the separation. We have a point in the recipe, sorry, let me go back to that. A point in the recipe that selects those spots, and those are highlighted in red, and then you take the selection and you basically spot heal inside of it, and it does that. So it just erases it out of the image, and here's now the image without those bright specks bothering you. So that was important. Might not seem important, but the ability to reliably find this Petri dish is critical. There's a lot of noise around here that can mess up your colony detection. So a point in the recipe reliably finds the inside of the Petri dish and says we're only going to look for colonies in there. Now in there, we start with our smart cluster step. It uses, if you're interested, k-means clustering. It's in our user manual to break up the grayscale image into a discrete number of grayscale pixels. So instead of 0 to 255, now you've got 0 to 3, or 1 to 3 if you choose 3 classes. Here we've got dark, medium, and bright. And this is a relative thing. So you've got dark, medium, and bright in that image. Dark, medium, and bright in the next image might be different originally, but they'll still be the same new dark, medium, and bright pixels after smart cluster. So it's a bit of a normalization step as well. So with smart cluster then we're able to select basically everything that's not dark, and that gave us our starting point to start finding the colonies. Now here's where I'm going to go into optimization, as we call it. I think sometimes there's still this notion that because we get into automated image analysis, we're somehow removing subjectivity. And I usually don't find that to be the case. I find it that automated image analysis lets more people be subjective more easily. Could be defining how much you dilate the features until you stop. Could be selecting the threshold value. You're still making a decision about where to call a feature. It's just instead of tracing, you're setting a threshold slider or you're setting a dilation slider. And that's just the way it is. We're still humans that are trying to add our own interpretation into a picture to get something out of it. We're never going to get away from that. That's the whole nature of the problem. What we can do is allow you to have some math and some defendable reasons behind why you drew the outline, where you drew the outline. I think it's a little easier to put into a publication. I drew the outline here because of an optimization of mutual information than it is because my grad student decided to draw the outline here. So that's what we had with optimization. What we did is we shrunk the initial colonies below where we knew their outline had to be. Now if you go into our dilation tool, yeah, you can set the dilation back to wherever you want. You set it at 20, there it is. You set it at 24, there it is. You set it at 28, there it is. I couldn't tell you which one of those is right. Either one of those could be where you should stop drawing the colonies. But if your measurement is going to be what's the diameter of the whole thing, or what's the extent of the filamentation, you set the dilation to give you whatever measurement you want. Publications hate that, I think. So what we can do is you can add that step with whatever setting you want, then you click on it in the recipe and you set optimize. And you give it a range of dilation to explore. See, I'm not sure what it should be. Let's try anywhere from 5 up to 30. You tell me where it should be, MIPAR. So that's what this looks like. What you see in front of you is exactly this. You get a movie of the dilation running over your range, and you get a corresponding plot of mutual information. Real simply, that is a measurement of how similar two images are. So it's measuring the mutual information between the dilated result and the original image. That's it. When it runs, it looks like this, and I'll loop back and forth because it plays quickly. It iteratively dilates from 5 to 30, and the software chooses the value that maximizes that value of mutual information. So once it's done, you accept that value, and that's the value of that step. Now, you can choose to lock that number in, so every time you run the recipe, it's going to select 24. If you don't lock it in, it's going to do this every time you run this recipe on a new image. It might come up with a different number. But that allows flexibility in dynamic steps that can adapt themselves to changing imaging conditions. Different experiments call for different approaches. Some experiments we like to lock it in, basically teach it on one image and run it on the rest, or let every image learn its own value this way. So that's optimization. We can run it on a number of different steps, the erosions, the dilations, the thresholds, all kinds of things. Water shedding, optimizing a water-shedded structure against an edge-brightened image can be a very powerful way to minimize the number of wrong separation lines that are drawn. We use the optimization for that. Now, once we have our optimized selection of the colonies, we look inside of them using a local threshold tool, and this lets the user set on a per separate feature basis the percentile bright pixels that they want to call the colonies. So I think this user used 70 percentile. They said take the top 70th percentile pixels inside of each colony and call that the center. It's not a global threshold across the whole image, it's a per colony threshold to define the center. Then I don't show it, but we did run optimization on that as well to have objective definitions of the center of the colonies. So at that point you had the whole thing, the centers, and that's all that you need. Because we've got very powerful Boolean operations. You can put images in and out of memory just like on a calculator. It's set it to M1, M2, M3, and you can recall those images at any point and do operations between all kinds of things. So you've got the building blocks now. We have total, center. You can use then logic operations to give you just the filaments, and with some other clever operations, the blue ones, the colonies free of filaments. So that's a breakdown of some of the critical tools that we use to solve this problem. What this user told us is that he built that in 10 minutes during my talk. He then spent about another 20 optimizing it to let it run on other images. And he was able to then batch apply this to several hundred of these in about 30 minutes. And this let him extract all the measurements he needed. It's almost obligatory these days to talk about machine learning around image analysis. So I wanted to do that, and afterwards I'll invite Paul up to start his bit. Question we get asked, do you do machine learning? Not yet? Then we have to explain why not. Why are we late? Quite honestly, we haven't needed it. We are solving with MIPAR's engines problems that we find other products are needing to resort now to some kind of machine learning to try to solve. We've been doing complicated things with recipes quite easily, and users have been. But it is coming because there is a value to it, and we truly believe. And we're going to do it just a bit differently. At its core, not really. Luciano gave yesterday, I thought, a fantastic talk on machine learning, one that I would have been willing to pay tuition to go to because it was a crash course in machine learning that I think every person around image analysis should attend. So thank you for that talk, and maybe we'll pay you in beers or something later. I don't need to explain a thing about what machine learning does or how it works. I'm just going to show you in principle how it's going to work when it goes into MIPAR. What it would look like is, as a first step in the recipe, the user would, as you've seen before, trace around what they're interested in. But here's where it's going to get a little different. What's troubled me with a lot of the machine learning solutions I've seen is that they take this tracing, you can trace different classes, and it spits out a color-coded segmentation that you have to swallow. You got to go with. And I've yet to see a machine learning segmentation that is the final solution. It feels almost like you're out stranded in the ocean and someone's thrown a life preserver that you can't quite get to. There's nothing you can do to get to it. Machine learning has spatial limitations, spatial resolution limitations, because as Luciano talked about, you're using a kernel around your pixels. So you've got challenges at boundaries between things. You have a phase contrast image. Yeah, your machine learning might give you the patches where your cells are, but it doesn't separate them properly. And maybe the outlines aren't as refined as they should be. Or maybe there's holes inside of your wound. Or maybe there's some other small features that you want to reject. But those are the easy things. The hard part is getting to the point where this image looks like what we all wished every image analysis problem would look like, what you care about lit up against everything else. If every image analysis problem looked like this, and all anybody ever had to do was threshold, fill holes, remove small objects, or sort however you want, that's a heck of a benefit. So that's what we want our machine learning to do, to let it transform any class of problems into the most approachable kinds of image analysis problems, where what you've selected is lit up against everything else. Now, if you selected multiple classes, you'll get different images with those things lit up and everything else not. Probably give you a classification map that shows which pixel has the highest lighting in the classes that you've selected. But it's going to take experience out of a lot of the recipe building. So it's going to make problems even faster to solve in MIPAR. Because I did this in MIPAR. This is a few steps, but it's the majority of the recipe to get it from here to here. Afterwards, it's pretty easy. If one step that anyone can do with a crayon gets you here, that's going to be big. So you're going to need even less experience to use it, but importantly, it's not going to compromise the flexibility. You're still going to be able to take the machine learning output, and process it however you want. Smooth it, separate it, fill the holes, get it to the final solution, instead of just accepting the machine learning output as what you got to go with. So that's coming. We're probably going to roll it into the base package, and we expect it out by the end of this quarter, the end of the summer, I think. So that's where I'm going to leave off. Paul's going to come up here now and talk about a few other life science applications that we saw with MIPAR, and then I will come up at the very end just to close it out.
So I am an application specialist at MIPAR. I specialize in life sciences, so if you guys are interested in using MIPAR for life sciences, chances are you and I will be working together to put together some of these solutions. So digital pathology is a field that benefits immensely from machine learning and being able to identify and diagnose different types of pathologies is incredibly powerful, but incredibly challenging. And this is why you need machine learning. Fortunately, a lot of times scientists are not really interested in this. What they're looking for is to look for differences between variables in their studies. So all we're trying to do is quantify things like cirrhosis. And in this case, what we have is a trichrome image. What we're looking to do is to segment and characterize the scar tissue in the pathology image. And in this case, the scar tissue is in blue. This is incredibly challenging, mostly because in order to properly quantify this, not only are you interested in the percent area of this, you're also interested in the characteristics of this scar tissue. So, challenges. You have biological variation. You have challenges in sample preparation. And lastly, we need a reliable characterization of this data. So this is a well-known, published method of stain deconvolution. So instead of separating an image based on their red, blue, and green channels, what you can do is you can actually separate these a stained image based on the stains. So in our case, we're separating it into our scar tissue, which was much darker in the image, and everything else is bright. And this solves 90% of the problem, because what you have now is I don't want to call it a machine learning solution, but what you have here is a two-step result of what you're interested in. So once we have this, what we can apply is grayscale clustering, which what John described. What it does is it separates our original image into a number of classes. From here, we can select this number of classes consistently every time to give us an objective threshold. Once we have this, within a couple of steps, what we can do is we clean this up, and what we see in green is what is identified as scar tissue. So this is 4 steps, and we can make this much more complex. If we see variation in this, we can make up for it to help you make this more robust. As John mentioned before, your measurements are only as reliable as your segmentation. So from here, once we have our mask, which is basically everything in green was a binary mask, we can generate any number of measurements on this. If you're interested in a global area fraction, you have it there. If you're interested in the local thickness, we have the distribution profile of that image as well, and then a local area fraction. So why is this important to us? So if you're interested in simply quantifying this and your project is done, this is fantastic. You can simply use this as fully automated, run it as a batch, get your results, and you have your comparison study. A further application is what you have now done is reliably segmented the scar tissue, which is what's primarily used to diagnose cirrhosis. You can use this to train your neural network and reliably create a diagnostic tool. We're not in the business of doing this. We're in the business of enabling this technology. Next application I want to talk about is co-localization in confocal fluorescent studies. In this specific case, what we're interested in is a colocalization of protein 1, which is our general green stain, protein 2 puncta, and then we want to generate these measurements per nuclei. Some of the challenges that we face is background non-specific signal. We have artifact particles which are not localized to the nucleus. We want to remove those. And then lastly, we just have some unseparated nuclei that we should take care of. So how do we do this in MIPAR? So we take a very logical approach to all image analysis problems. What we try to enable is if a user sees their way to a solution, we provide the tools for them to do exactly what they're trying to do. In this specific case, first thing we're going to do is we're going to identify a region of interest. This is pretty fundamental to image analysis. What you want to do is we know that all of our information is going to be localized to the nucleus and within the nucleus, so why not just find a nucleus to begin with? So within 7 steps, and I can describe these fairly easily, we can identify our nuclei. So let's take a look at this. Our very first step is we identify the channel where our nucleus data is, and in this case, it's in our blue channel. We apply a noise reduction filter and the basic thresholding to select the nuclei. What we find is that it's going to have holes in it, it's going to have some noise around it. We do some cleanup steps and a separation step to make sure that our nuclei are separated. Once we have this, we remove any partial nuclei because we don't want to be generating data on partial regions of interest. Next thing we want to do is we want to identify the puncta in protein 2. Again, fairly simple. We know that this data is in our red channel. We do a noise reduction, an adaptive threshold. It's pretty popular. It works similar to how your eyes work, where it looks for local differences in intensity instead of global ones. And then we clean this up to give us a reliable selection of what we identify as protein puncta. Lastly, we know that our protein 1 is going to be localized to our nuclei, so why not just use our nuclei mask as our region of interest for that? So without even trying to segment this green channel, we know that this stain is going to be within the nucleus, so we can use that information to our benefit. So how do we set up the measurements for this? So it's fairly intuitive. All we're doing is bringing up a layer that we have already created. We set this as a companion layer, which just means it's a layer of interest. It's a step of interest to us. We're going to be using this step to generate other measurements. Then we call back our nucleus region of interest, and then we do this again for our general protein 1 stain. So what does that result in? What we can do is very nicely demonstrate and visualize your intensity profiles as well as your number of puncta per nucleus. So this is very nice to see immediate feedback of your data. And this recipe is fully automated. So you just load your images in, click run with the recipe, and you can walk away. There's no user input necessary for the measurement generation or the segmentation. A nice feature that we have is being able to kind of create our custom measurements for this. And this is kind of a prototyping environment where you're looking for trends in your data. In this specific case, what we're looking for is correlating this intensity of protein 1 to the number of puncta in protein 2. So why not just take the protein 1 intensity and divide by the protein 2 count? And what we can see is a very strong relationship between those two immediately. And from there, we can use that as a way to, a guide to processing this data further. And if you have spatial information, if for some reason this was changing from left to right, if you were getting more intensities from left to right, you would very easily see this in this visualization. So what we have demonstrated here is a reliable, repeatable way to do colocalization. It's fully automated. A broader application, this is what we have used this already for, is an affordable high throughput drug screening for a fluorescence imaging. So we work with an existing high throughput system. They create absurd amounts of data, and we're able to do is process it effectively and give them the data they're interested in seeing. The last thing I wanted to talk about is the data set that was provided for analysis, which is the nematode three-dimensional data set. And for this project, and as far as we understood, what we're looking for is the intake of gold nanoparticles localized to the internal of this nematode. So what we're interested in is count these particles and measure the volume of them, which are localized internally to the nematode. Analysis challenges. This data set is absolutely massive. It took me longer to download it and to screen it than it took me to process it. There is presence of imaging artifacts, so things that make it challenging to fully automate. And then we are working in a third dimension, so that we need to be able to reliably separate these features in 3D. Because MIPAR does our two-dimensional analysis so well, I know that that's subjective, what we like to do is we take three-dimensional problems, we break them down in 2 dimensions, and then we reconstruct this into 3 dimensions to give us our measurements. So this is our 2D slice. I took a portion of this because for the sake of demonstration, I was just trying to derive an example to demonstrate. I didn't want to process the entire data set. So this is a 2D slice through the sample, and what we're looking to do here is to remove the imaging artifact, which is this white line on the right. We want to identify the region of interest, which is our cross-section. We want to identify the nanoparticles, which I will show later on. And then we want to segment these artifacts. So artifact identification is fairly simple here. It's a very obvious artifact. It's a very large line that is running through the right side of our image. And what we want to do is we want to remove this. Because if we don't, what's going to happen is, it's going to sound overcomplicated, but what it does is it's warping the pixel space. So when you're comparing pixels to each other, all of a sudden, all the pixels around that white line are darker than they're supposed to be. I'm relying on the contrast of my cross-section to be darker than its background. Now all of a sudden my background is darker than this artifact. For this reason, that needs to be removed. And it's present in 10 slices, which is, it's not significant. There might be some nanoparticles in those 10 slices that I would miss if I were just simply take that data and remove it. So what we do is we identify our region of interest, and we use a grayscale. I guess I don't want to try and explain that, but it's removed from our images. And if there is no artifact present, then it's not executed. So that rate limiting step, which takes quite a bit of computation, is not executed at all. So next we identify a region of interest, and this is fairly simple. Again, looking for features that are darker than their neighbors, and it's selecting it reliably independent of the morphology of the cross-section. Nanoparticles. So the nanoparticles in this case are very bright foci because they're gold, and SEM image, when it's majority carbon, the gold nanoparticle is going to light up very nicely. So this is not complicated. You just select that using a basic threshold. What we did though is used our region of interest to make sure that anything that's being selected is to the interior of our cross-section and not outside of it, just to make sure that we're not picking up any small artifacts that we might have missed during pre-processing. So, two-dimensional requirements, we meet all of those. So, because we did everything well in 2 dimensions, our resulting reconstruction is automated, accurate, and it's efficient. It's effective. So, we have a 3D toolbox which we primarily use for FIB-SEM data sets, which lets us kind of reconstruct our 2-dimensional data sets to generate measurements in 3D. We did find that after measuring these nanoparticles, they're within the specification that was in the publication. I just visualized 2 of these. I think there was 5 total in that small section, but they were spaced out so far away it would not have been possible to visualize them nicely. And these nice visualizations can be exported directly into Aviso to create very nice videos for conference talks and professional presentations. So to review this, we've demonstrated a robust method to quantifying these large data sets in the presence of artifacts to identify our features of interest. And this approach can be used to quantify all sorts of things. This is how we do three-dimensional applications. And yeah, I think this is all I had for now. Some couple of closing things I want to say is that we work very hard to onboarding new users. We understand that not everybody's an image analysis experts, and this is why you have us, and we really help you in these early stages to put together these recipes and teach you what we did and why we did it. That way, we make you an expert in your own specific field of image analysis instead of leaving you to fend for yourself, trying to solve oftentimes very complex problems. I'm going to hand it back to John.
Thank you, Paul, for those case studies. He's a heck of an asset to the team, and he does work, I can attest to, very tirelessly with users across all levels of expertise to help them get the most out of the product. So it's great to have him on the team. So I just wanted to leave you with a couple of very small takeaways. I want to go back to our Venn diagram where we started of how we see MIPAR. And it really is a very powerful balance between flexibility, efficiency, which enables support. Enables us to give it, enables users within your facility to give it. Truthfully, we are solving problems automatically that other solutions have struggled with. That has garnered a lot of the users that have adopted us already. All of the rest of them we're solving more efficiently and faster. And again, the combination of those two things lets a private business offer hands-on custom solutions to people when they need it, because we've built ourselves a tool that lets us do those problem solutions sometimes in 10 minutes. So come visit us. We're out there for the rest of the day. Our website is MIPAR.us. On that website, you can do all kinds of things. We have a 30-day trial. We have a recipe store that's always growing. We've got 50 pre-built recipes, 50 plus, I think, recipes across all application areas. It's broken down, it's searchable, it's visual. You can go into the life science section and see different recipes for those applications. You can download the recipe, the example image, drag them in, see how it works, run it on your image, tinker with it. It's a powerful resource, and they're free. And come bring your images. Bring them outside to the table. Submit them through our page on the site. mypar.us/try-image, we get hundreds of submissions of images of people with problems. They give us the image, they describe it, hopefully they give us an annotated result, what they want found, and we work hard to put together custom recipes for them anywhere between 3 minutes to an hour. And we'll send you back your recipe, usually with a demo video explaining how it works, showing you how it runs, how you make measurements from it. Also on the site, of course, we've got our user manual, we've got tutorials, we've got past webinars that have been recorded, and we're trying to hold webinars, public ones, more frequently. Web-based ones, we'll send out invitations to anyone on our active user list and probably plug it on social media. We get good attendance at the webinars, and people, we solve real problems in front of them using the tool. They're great experiences. So with that being said, I did want to mention that we are offering aggressive programs to get MIPAR on entire campuses, entire core facilities. We know, because users have told us, that they solve more problems more quickly than with open source solutions. But it's hard, you've got to overcome the barrier of the open source thing. So we are sensitive to that, and we want to do what we can to get all 30,000, I don't know how many students there are at UMB, lifetime access to MIPAR, students, faculty, staff, researchers, as long as they're here. We're trying to do that across as many universities as one. There's also a serious education benefit to it. Obviously, the research problem-solving side is huge, but the educational value of being able to click on each step and see what it did, to play around with these algorithms, people that have adopted it in image analysis courses love it. So we want it on campuses and core facilities for that reason as well. So if you're interested, talk to us. We are really trying to offer aggressive site-wide license options as best we can. That's all that I had. Again, we're going to be outside all of today. If you have images, bring them by. We will do our best to make a start-to-finish solution for you on the spot. Again, my name is John Sosa. I'm with MIPAR. That was Paul Sewell, our Senior Application Specialist. Thank you very much for your attention, and we've got some time for questions if there are any. Thank you.